Welcome to AP Biology. Today I want to talk about receptor tyrosine kinases. So these are receptors that you'll find in the cell membrane of cells, and they have an outer part here that's outside of the cell. We would call that the ligand binding domain. Because it's the part of the protein that accepts the ligand, and so it has to be just the right shape for this particular ligand, so it's a very specific fit. So if this is serotonin, um, or let's say, yeah, if this is serotonin, then this receptor would only fit with serotonin. Or if this is the epinephrine molecule, it would only fit with epinephrine. Okay, and then there's this part here, which I would call the intramembranous domain. And so what you should keep in mind with that is that these need to be hydrophobic. They need to have this part of the protein needs to have amino acids that have hydrophobic R groups because they're going to be snuggled up next to the lipid on all sides. And so if they're not hydrophobic, then they would be repelled by the lipid bilayer. And so they wouldn't be able to fit into the cell membrane. And then we have this inside part. We're going to call this the cytoplasmic domain. So we have these three parts, and so now what I want to talk about is how specifically this receptor tyrosine kinase is different from other receptor proteins. So these are membrane proteins that attach phosphates to tyrosines. So this is a type of amino acid. So there are several amino acids, right? There are 20 of them, and some of them are able to be phosphorylated more easily than others. And so if you remember what phosphorylation means, phosphorylation means you can put a phosphate group onto something. And so you can see this one actually already phosphorylated. That means it's already activated. It's not always activated, but if this ligand has bonded in here, if there's some binding there, some affinity, then the enzyme will be phosphorylated. So tyrosine is just an amino acid that can be easily phosphorylated. This word kinase, that's part of the name here, right? Receptor tyrosine kinase. So this is a receptor that it's a protein, so it's made out of a whole bunch of amino acids. One amino acid, tyrosine, is found in here, and it's a special one because it can be phosphorylated. Okay, kinase. You can see from the name, kinase, ACE, means it's an enzyme. What does a kinase do? It catalyzes the transfer of a phosphate group from ATP to some other molecule. So this is one example of a kinase molecule. There are many, many other examples. It's not that it gets phosphorylated itself, it's that it can go ahead and phosphorylate something else. That's the key. That's what makes it a kinase. So we'll see this kinase right here, and then when we do the signal transduction pathway, we'll see that there are other kinases in the cell that will become important. So receptor tyrosine kinase can trigger multiple signal transduction pathways at once. So if this phosphate group phosphorylates one protein or enzyme, and if this one phosphorylates a different one, and if this one phosphorylates a different one, you can have a whole bunch of different um, pathways initiated at the same time. So let's say this is epinephrine and it binds to a receptor on the cardiac muscle cell. It might have one phosphate group that phosphorylates an enzyme that you need in order to stimulate the cells to contract harder. And another one might say, hey, let's speed this up. Let's stimulate the heart cells to pump faster. And then another one might stimulate glycogen to be broken down to glucose because we need more ATP when we pump a heart really hard. So there can be multiple pathways going on at once from just that one initial ligand. So here's what it looks like. You have a signaling molecule or a ligand attached right here to the receptor, and you'll actually have that happen to two of them. I hate that we call it a monomer here because that monomer sounds like it's an amino acid or a monosaccharide, right? But this is a different use of the term. It just means a protein that hasn't come together with its friend yet. And so here they are together with their friends. So if you have two signaling molecules and they attach to the receptors will come together, and this is a functional activated um, receptor tyrosine kinase. So once they're activated, you're going to need 6 ATPs to phosphorylate each of the tyrosines. So the fully activated receptor tyrosine kinase is completely phosphorylated. It's got those 6 phosphate groups on them. Once it's activated, then it can go ahead and phosphorylate other molecules. So this one is phosphorylating this molecule, this one is phosphorylating that molecule. Once they're phosphorylated, they are activated, and they can go ahead and cause different cellular responses. Maybe this one says, hey, contract harder, heart, and this one says, hey, heart, I need more glucose. You better break down some glycogen to make glucose. So you can have different cellular responses based on just the binding of epinephrine to this molecule. So here's the whole thing. This is when it's inactive. They are apart. Once you get two signaling molecules, the two proteins come together to make one larger protein, and it gets activated. And then you phosphorylate, use ATP to phosphorylate each of these tyrosines. And then the kinase part is that you're phosphorylating other proteins. So these are going to get the phosphate groups and go on to catalyze some other reaction.